Hello friends, in this video we are going to discuss about translation in prokaryotes. We will discuss this in series of videos. In this we are going to discuss about its introduction. We will discuss about definition of translation, then central dogma, then we will discuss about components of translation, we will discuss about ribosome in detail, then we will discuss about initiation factors IF1, IF2 and IF3. We will discuss about synthesis of amino acyl transfer RNA. Under this, we will discuss about its two-step synthesis process and we will also discuss about its importance. So stay tuned. Translation in prokaryotes. Let us see its introduction. Representative prokaryotes E. coli. So first of all, what is translation or what is protein synthesis? The process by which a cell makes proteins using the genetic information carried in messenger RNA. This messenger RNA is made by copying DNA and the information it carries tells the cell how to link amino acid together to form proteins. In prokaryotes, it is a tightly regulated process that begins with the assembly of initiation complex. This initiation complex we will discuss later. Now the central dogma. It is a theory stating that genetic information flows only in one direction. DNA molecule makes its copy by the process of replication. DNA then forms RNA by the process of transcription and RNA then forms protein by the process of translation. So here the information from DNA to RNA to protein flows unidirectionally. There is no complementarity between amino acid and messenger RNA. Hence, translation is not controlled by complementarity but by genetic code. Genetic code we have already discussed in detail in separate video, the link of which is given in the description box. Since amino acid cannot read this genetic code, they are dependent on an adapter molecule called transfer RNA. We have already discussed transfer RNA in separate video. You can refer my video, the link is given in the description box. Now let us see the components of translation. First is messenger RNA. It acts as a template. It carries the genetic information copied from DNA in the form of a series of 3 base code words, each of which specifies a particular amino acid. Therefore, codons specify amino acids. Second is transfer RNA, which is an adapter molecule. It has anticodon for codons of messenger RNA, and these codons are complementary to anticodons. Hence, base pairing occurs between these two, that is, anticodon and its complementary codons. Hence, it brings accurate or specific amino acid in accordance with the codon. It deciphers code in messenger RNA, or in other words, it discovers code in messenger RNA. Each type of amino acid has its own type of transfer RNA, which binds it and carries it to the growing end of a polypeptide chain or protein. Then enzymes. Among enzymes, first is peptidyl transferase. It facilitates the formation of peptide bonds between amino acids. It is a component of large ribosomal subunit. Second is amino acyl transfer RNA synthetase. It attaches appropriate amino acid to its transfer RNA, that is, loads or charges transfer RNA. Then third is methionine transfer RNA formyl transferase. It catalyzes the addition of formyl group to methionine. So formyl methionine is the first amino acid of protein or polypeptide chain in prokaryotes. Then ribosome. It is manufacturing unit. It has two subunits, small subunit and larger subunit. Small subunit reads messenger RNA and larger subunit joins amino acid to form polypeptide. Now let us discuss about some of the important aspects of ribosome. Ribosome normally exists as separate subunits. that is smaller subunit and larger subunit. They come together when bind to a messenger RNA near its 5' end. Then it reads the nucleotide sequence of messenger RNA from 5' to 3' direction. It synthesizes protein from amino acid in an N-terminal to C-terminal, that is from amino terminal to carboxy terminal. Now the location of ribosome, it is located in the cytosol, either free floating or associated with endoplasmic reticulum. Now the ribosomal sites. First is amino acyl transfer RNA binding site or A site. It is the entry site for new transfer RNA charged with amino acid or amino acyl transfer RNA. The peptidal transfer RNA binding site or P site, it is occupied by peptidal transfer RNA or binding site for transfer RNA that carries the growing polypeptide chain. Third is the exit site or E site. It is the exit site for the deacylated transfer RNA, or in other words, it is exit site for the transfer RNA which has done delivering the amino acid. Hence, it is a binding site for transfer RNA following its role in translation and prior to its release from the ribosome. Now, let us see the structure of the functional ribosome. This is smaller subunit, this is larger subunit. This is the messenger RNA. Three sites of ribosome are amino acyl transfer RNA binding site or A site. This is peptidal transfer RNA binding site or P site, and this is exit site or E site. Fifth component of translation are proteins. These are soluble factors that are required for proper initiation, elongation and termination of translation. Here we will discuss about factors involved in initiation. Factors of elongation and termination we will discuss in later videos. Initiation factors. Initiation factors in prokaryotes involves coordinated action of IF1, IF2 and IF3, known as initiation factor 1, 2 and 3. Their main function is to assemble the ribosomal subunit around the messenger RNA and recruit initiator transfer RNA. IF1 prevent the premature entry of transfer RNA into A site of ribosome. IF3 prevent premature association of ribosome subunits. Hence, small ribosomal subunit is separated from large subunit with the help of IF1 and IF3. Or in other words, its role is to stop the 30th subunit binding to 50th subunit in the absence of messenger RNA and initiator transfer RNA. Otherwise, their association will result in non-functional ribosome. IF2. It has ribosome-dependent GTPase activity, which regulates and directs the initiator transfer RNA to its correct position in the initiation complex. Initiator transfer RNA is the transfer RNA charged with N-formyl methionine amino acid. When specific amino acid bind to its specific transfer RNA, it is known as amino acyl transfer RNA. Let us see the synthesis of amino acyl transfer RNA. It is two-step reaction. First, reaction of amino acid and ATP to form an amino acyl adenylate or amino acyl AMP. Second, amino acyl group of amino acyl AMP is transferred to 3' end of transfer RNA to form amino acyl transfer RNA. So the overall reaction is amino acid plus ATP plus transfer RNA in the presence of enzyme amino acyl transfer RNA synthetase gives amino acyl transfer RNA plus AMP plus inorganic phosphate. Now let us see the importance of amino acyl transfer RNA synthesis. To take part in protein synthesis, each amino acid must be covalently linked to a transfer RNA molecule. This depends upon the adaptive function of transfer RNA to ensure that correct amino acids are incorporated. So, in order to take part in protein synthesis, amino acid is covalently linked to transfer RNA. So, the transfer RNA function as adapter molecule and correct amino acid is incorporated. Covalent bond between amino acid and transfer RNA is a high energy bond, and this enables the amino acid to react with the end of the growing polypeptide chain to form new peptide bond. Hence, synthesis of amino acyl transfer RNA is also referred to as amino acid activation. So this is all for today's video. If you like this video, please hit the like button, share it and subscribe my channel. Thank you.